The true value of the studies undertaken by John Snow and Ignaz Semmelweis were only recognised with the advancement of the germ theory of infectious diseases. Girolamo Fracastoro, who lived from 1478 to 1553, proposed that epidemics were caused by spores that could be transmitting infection, although he considered these to be of a chemical nature rather than being biological agents. Another Italian, Agostino Bassi, who lived from 1773 to 1856, was the first person to associate an infectious disease with a microorganism. He identified a fungus as the cause of muscadine, an epidemic of silkworms. Antony van Leeuwenhoek, who lived from 1632 to 1723, had observed minute organisms using his microscope. For many years, however, these minute life forms were considered to generate themselves spontaneously. Louis Pasteur pioneered work on fermentations and demonstrated that spontaneous generation of microscopic life did not occur. Ultimately, Robert Koch established a series of tests used to assess the nature of the agents of infections, Koch's postulates, published in 1890. Prior to this, cholera was thought to spread through the agency of a miasma, or bad air, containing decomposed matter known as miasmata. These were considered to be the cause of epidemic illnesses such as plague and cholera. According to the miasma theory, victims developed the disease after inhalation of miasmata. There was no place for the person-to-person spread that we now know is responsible for the transmission of infections, the contagion theory of disease. We now recognise that cholera is caused when people drink water contaminated with Vibrio cholerae derived from human feces. Vibrio cholerae is a pathogen found only in association with humans. Like typhoid, there is no animal reservoir for this infection. The incubation period for cholera is between 1 and 4 days, and the infectious dose is very high. It has been estimated that it requires a dose of 10 to the power 10 bacteria to initiate infection. That's 10 with 10 zeros after it. Symptoms of cholera include fever, vomiting, and profuse watery diarrhea. As the disease progresses, the patient's stools take on the appearance of water in which rice has been boiled, hence the description of rice water stools. Healthy adults typically lose about 2.5 litres of fluid a day. In cholera victims, the fluid loss can amount to between 10 and 15 litres per day. This is a consequence of the action of cholera toxin, also known as choleragen. Not all isolates of Vibrio cholerae produce cholera toxin. It is only those bacteria that have themselves been infected with a bacteriophage or bacterial virus, known as CTX-F or CTX-Phi, are toxigenic. The toxin is a classical bacterial A plus B toxin, comprising 1 A subunit and 5 B subunits. The B subunits form a pentamer structure that binds to the gut epithelial cell surface, allowing the toxin to enter the cell. There, a portion of the A subunit is released, and it acts as an enzyme that catalyzes the formation of cyclic AMP. This is an important signaling molecule in cells, activating many proteins. One of the proteins that is activated by high levels of cyclic AMP is the protein responsible for exporting sodium ions from the cell, causing a disturbance of the body's electrolyte balance. To balance the loss of sodium ions from inside the cells, chloride ions move out of the cell, generating a strong solution of saline in the lumen of the gut, and this causes water loss from the body as a result of osmosis. This results in circulatory collapse and in death. The blood takes on a tarry appearance as the water content reduces. This means that organs are not receiving adequate oxygen. The skin of the patient takes on a bluish hue, referred to as cyanosis, and the lips turn blue and then black as a result of lack of oxygen. Failure of the blood circulation causes the victim to suffer severe cramps, and in extreme cases, gangrene may set in. Untreated, cholera has a mortality rate of about 50%. Antibiotic treatment is not required in most cases, although in very severe cases, tetracycline may be used. Successful treatment is through rehydration. In mild cases, this can be used by oral rehydration therapy using a simple solution of salts and sugar, including potassium as well as sodium chloride. Although this may be effective if given orally, it is much more effective if it is delivered via an intravenous drip. In severe cases, intravenous dextrosaline drips are used. Remarkably, histological examination of the gut from fatal cases of cholera shows a perfectly normal cellular appearance. Cholera toxin causes no apparent damage to the gut at a cellular level, and yet, because of its effects on electrolyte balance of the body, its effects can be devastating. Now let's turn to bacillary dysentery. Cholera is a potentially fatal infection that results from minimal interaction between the pathogenic bacteria and their victim. The toxin only acts on the cells that line the gut. Bacillary dysentery represents a more invasive illness. It's caused by bacteria of the genus Shigella. Clinically, it manifests itself as a bloody flux, diarrhea typified by the presence of copious blood, pus and mucus in the stools. There is also a severe abdominal pain associated with the condition. The most severe form of the disease is caused by Shigella dysenteriae, and this species is particularly prevalent in the Third World, where a high mortality rate is found among untreated patients. In the United Kingdom, most cases are caused by Shigella sonni. This causes a mild dysentery. Shigella sonni is unusual in that it can ferment lactose, but only after a prolonged incubation period. Two other species, Shigella flexneri and Shigella boydii, cause disease of an intermediate severity, more serious than caused by Shigella sonni, but less severe than by Shigella dysenteriae. In the case of all Shigellas, infection results in the invasion of the tissues of the gut mucosa. Inflammation is, however, confined to the submucosa, and it doesn't spread beyond this region. Shigella dysenteriae elaborates a toxin known as the Shiga toxin. Like cholera toxin, it is encoded by bacteriophage DNA. Bacteria are viruses that infect bacteria. Temperate bacteriophage do not kill their bacterial hosts immediately, and they integrate their genomes into the host bacterial DNA. The toxin has neurotoxic properties, and it is thought to be responsible for inducing coma in fatal cases of dysentery. In the United Kingdom, cases of Shigella sonni dysentery are most frequently seen in nurseries and play schools, although they often also occur in psychogeriatric institutions. The disease is spread because of poor personal hygiene, particularly observed by small children and psychogeriatric patients. The problem is exacerbated if the staff in such institutions are overstretched. Infections under these circumstances probably spreads as a direct consequence of direct fecal-oral contact, often via soiled fingers. Small children love to suck their fingers but may not be very careful about washing their hands after going to the toilet. Shigellas can readily pass through toilet paper. The infectious dose of Shigellas required to initiate dysentery is very small, being of the order of 100 to 1,000 bacteria. This facilitates the spread of the infection in such situations as nurseries. So now let's consider enteric fevers. Enteric fever is a collective term given to the invasive infections caused by Salmonella typhi, the cause of typhoid fever, but also by strains of Salmonella paratyphi that cause paratyphoid fever. This infection represents the ultimate in penetration of a host by a pathogen, as the bacteria gain access to the body via the gut and then go on to cause a systemic generalized infection affecting many organs of the body. Salmonella typhi cells can survive inside white blood cells that usually defend us from such infections. In enteric fevers, these cells become vectors carrying the pathogen around the entire body. Like Vibrio cholerae, Salmonella typhi is a pathogen that only has humans as its natural host. The incubation period of typhoid is generally about 2 weeks, and the infectious dose is about 100,000 to 1 million bacteria. This dose may be lower if the patient has been taking antibiotics when exposed to the infectious agent. During the incubation period, the bacterium colonizes the small intestine. From there, it enters the lymphoid tissue, and thence it reaches the bloodstream. The bacterium also tends to localize in the spleen, liver, and gallbladder. When the bacterium enters the bloodstream, it causes septicemia, blood poisoning. The first clinical symptom is fever, and this coincides with the onset of septicemia. During the early stages of the clinical disease, patients suffer from a dry cough, headache, and abdominal pain. Constipation is a very common symptom during the early stages of typhoid fever. As the disease progresses, areas of inflammation occur in localized lymphoid tissues known as Peyer's patches. After about 2 weeks, patients develop diarrhea. At about this time, rose spots appear on the patient's abdomen as a result of subcutaneous hemorrhage. Later complications of the disease include hemorrhage, intestinal perforation, osteomyelitis, and meningitis. Untreated cases of typhoid are associated with a mortality of about 10%. The most severe cases are caused by bacteria that carry a VI antigen, capsular material that affords protection from the body's immune defences. Salmonella typhi is an intracellular parasite, and the capsule also provides protection from the immune response, as the organism is hidden inside macrophage cells. About 5% of typhoid patients become asymptomatic carriers, often harbouring the bacterium in their gallbladder. A small number of carriers excrete Salmonella typhi in their urine. Water is the principal vector of typhoid fever, but this need not necessarily be in the form of drinking water. Ice used in drinks and the water used to wash salad, fruit and vegetables have been found to cause typhoid. The symptoms of paratyphoid fever are similar to those of typhoid, but Salmonella paratyphi is associated with a milder disease and causes fewer deaths.